So, good afternoon to all. I, Dr. Vivek Asati, Associate Professor, Department of Pharmaceutical Chemistry, on behalf of ISF College of Pharmacy, Moga, Punjab. Welcome to respected Director Dr. G.D. Gupta sir, ISF College of Pharmacy, Moga, Dr. R.K. Narang sir, Vice Principal, our speaker expert, Dr. Piyush Ghode sir. So, I welcome to Dr. Piyush Ghode sir in ISF Dialogue Series. Expert talk on molecular docking, a pivotal technique in structural-based drug design. So, Dr. Piyush Ghode, currently working as Assistant Professor in Department of Pharmaceutical Chemistry, School of Pharmacy and Technology Management, SVKM, Shirpur. He completed his B.Pharm from GYR Institute of Pharmacy, Moraban, Khargone, and M.Pharm from College of Pharmacy, IPS Academy, Indore. He completed his PhD from Guru Ghasidas Vishwavidyalaya, Bilaspur. He has more than 10 years of experience in teaching in pharmaceutical chemistry. He has published more than 10 papers in national and international journals of reputed and presented in many national and international conferences and won prizes. His area of interest is computer-aided de novo drug design. He has conducted various training programs on computer-aided drug design. So now I request to Dr. Piyush Ghode sir, please start your expert talk.
Okay, so thank you, Dr. Vivek Asati, and thank you, Dr. G.D. Gupta sir, for inviting me at ISFCP Expert Dialogues. And we'll begin with our topic today, and that is molecular docking, a pivotal technique in structure-based drug design. So I have designed this presentation in such a manner that even a beginner will be able to understand what molecular docking is and how we can proceed about performing molecular docking. So after completion of this session, we will be able to understand what is computer-aided drug design, what is structure-based drug design, different types of structure-based drug design, molecular docking, a typical docking result, methodology for molecular docking, and virtual screening. So as we know that molecular docking is a part of computer-aided drug design, we have to appreciate the field of computer-aided drug design that in the previous 1 to 2 decades, it has completely radicalized the drug discovery process. And why so? Because it provides industry an opportunity. What opportunity? To check upon two of the most fundamental assets for humankind in general and business in particular, and that is time and money. And if you just remember the phrase, time is money, it means it becomes money and money, that is money square. So pharmaceutical industry, it just focuses on computer-aided drug design so that it can save time, it can save money, and in order this money can be transferred to the welfare of the patients as well. So computer-aided drug design basically spans over a large area of drug discovery process. We can see that from target identification to preclinical tests, computer-aided drug design plays a pivotal role. So whereas in target identification and target validation, we can see that they are majorly the domains of bioinformatics, but the lead discovery, lead optimization, and preclinical tests, they are of more interest to the medicinal chemists. And the lead discovery involves library design, docking and scoring, de novo design, and pharmacophore. And lead optimization involves QSAR, structure-based optimization, and preclinical tests involve in silico ADMET and PBPK predictions. So computer-aided drug design methods can be further divided into two main categories. The first is structure-based and the second is ligand-based. So structure-based drug design is applied when we know the structure of the target. For example, if I talk about the target dipeptidyl peptidase 4, DPP4, which we are going to discuss to length in the coming slides, this target is well known, its experimental structure is deduced, and therefore, if you want to generate a new drug, you can take this target, you can take this its active site, and then you can design a compound which is interacting with it. So this is structure based. What methods can be applied in structure based drug design? They are molecular docking, molecular dynamics, de novo design, and pharmacophore. Whereas the other technique is ligand based design, where the target structure is not known. Say, for example, if there is a disease, for example, an orphan disease for which the target is not known, now you have some agents which are able to act and control that situation. So what you have the ligands structure. Now you use this ligands structure to extract some properties. You create some descriptors out of those properties, and then you correlate those descriptors to the activity of that molecule, which is called as quantitative structure activity relationships, or QSAR. And the other method is again pharmacophore. We can see that pharmacophore is coming at both the places, structure based and ligand based. So here pharmacophore is considered as a three-dimensional arrangement of some specific features which are responsible for the activity of a molecule. Now, these three-dimensional features can be considered as the interactions with the receptor, or they can be considered as some of the descriptors. So, in structure-based drug design, in a nutshell, we can understand through this slide, the molecular targets are known. What we do is we try to find out some ligands through ligand modeling, utilizing the active sites of these molecular targets, and then we go for experimental evaluation. Now, in this experimental evaluation, it might be possible that we may come across some of the aspects that can further modify or improve the properties of these ligands. So we go for lead optimization again in silico, and then this cycle can continue up to the point where you can get a good lead or a good drug candidate. So this slide explains how we can understand structure-based drug design. So we have a receptor, as we discussed. This is the example of dipeptidyl peptidase 4. It is an enzyme which is a target for diabetes management, and we can see a ligand sitagliptin here, which is a marketed drug against dipeptidyl peptidase 4. So what we expect in structure-based drug design, we expect that the drug or the ligand, it will be able to be accommodated into the active site of the ligand of the receptor in such a manner that it interacts with the specific amino acids of interest. Okay, so now let us come to this point. What can be the specific amino acids of interest? Here we can see again three-dimensional structure of DPP4 enzyme, and in magenta you see the active site of this enzyme. Okay, so in this active site there are three amino acid residues, namely serine 630, tyrosine 662, and tyrosine 710. These three are responsible for the activity of this DPP4 enzyme. Now, through site-directed mutagenesis, it was also found that glutamic acid 205 and 206, these are not necessarily under the active site, but these are called as extended active site. What they do is, actually, if you are able to block these amino acids as well, you will be able to block the activity of the DPP4 enzyme. So what we can see here is this yellow colored ligand is sitagliptin, and it is interacting with these magenta amino acids. The interactions are with glutamic acid 205, glutamic acid 206, and tyrosine 662. Now, what these interactions can be, again, we'll go step by step. So we have discussed about interactions. What kind of interactions can be there? So with glutamic acid 205, 206, you can see that there are these hydrogen bonding interactions. So these are some of the non-bonding interactions that can be possible in a drug receptor complex. With tyrosine 662, this hydrogen bonding interaction is available, but again, besides this, we have one more interaction. What kind of interaction is it? The same kind of interaction we can see here. So these are called as pi stacking interactions. Okay, so what we want is the interaction of amino acid residues which are important for the activity with the ligand, so that the activity of the receptor can be modulated. So for studying these kinds of interactions, which techniques are available? The first technique is molecular docking, which is basically the computational prediction of the binding mode and binding score of a molecular complex. Then we have molecular dynamics, which is basically a computer simulation method for studying the physical movements of atoms and molecules. Now, if you want to understand the difference between docking and dynamics, we can just think of a photo, a picture, and a movie. So if I talk about docking, docking represents a picture and dynamics represents a movie because it is movable in nature. The third way is de novo design, in which you have the active site of the receptor, and in that active site, you grow the ligands from scratch. That is why it is called as de novo design. You have the properties of amino acids with you. Now you make the ligands which are complementary in the properties to those amino acids, and in this way, you can design some new ligands. And then we have the structure based pharmacophore, which will be an abstract description of molecular features that are necessary for the ligand receptor binding. Now, among these, we are going to focus on molecular docking. So, in structure based drug design, what we want to show here is that there are total 4 techniques, among which the 2 techniques, molecular docking and molecular dynamics, these two are being able to be leveraged more by the researchers community, and we can see that the year-wise publications available in PubMed are growing for both the techniques. But we can see that the molecular docking is outperforming molecular dynamics by a large uh, margin. Why so? Because molecular docking is a technique which can be very easily performed on the computers. Those are available in the hands of students or the researchers. Whereas molecular dynamics, they would need some more high speed computers, which can not be available to many of the researchers, and therefore, specifically in the developing and underdeveloped countries. So therefore, molecular docking becomes a method of choice for many researchers, which can be utilized for a good research. So there are some terminologies related to molecular docking. The first is protein data bank. Protein data bank is a repository of the structures which have been experimentally deduced. Now there is a new paradigm in the previous 1 year, and that is AlphaFold. AlphaFold, if you remember, is an artificial intelligence technique through which we can predict the 3-dimensional structures of the proteins. So nowadays, protein data bank accommodates those structures as well. Then there is this crystallographic ligand. Crystallographic ligand is the ligand which is found in the experimental crystal structures in a certain conformation. We will see how this can be very important for us when we are performing molecular docking. Then we have a docked ligand. So the ligand pose which we will generate after the docking experiment has been done. And then we have the interactions as we have discussed on the previous slide. So how would a successful docking look like? This picture represents 2 ligands here. One in magenta is the crystallographic ligand, which means that it was present in the crystal structure that we have retrieved from the protein data bank. And the green one, it has been docked. So you can see that the docked ligand is having a very good overlap with the crystallographic ligand. And this is what an ideal docking would look like, where the interactions can be predicted with 100% certainty. And the other thing here to note is the score, which is minus 12.3 kilocalories per mole. Here it is showing the binding energy, that is the free energy of binding, and the negative sign, it represents that it is a spontaneous process. Okay, so if the process is not spontaneous, it means we have to provide the energy from outside, which is not typical, which is not possible in a typical biological setting. So therefore, process has to be spontaneous, and this negative sign is mentioning that. So as we have discussed interactions as the major part of molecular docking, so there are majorly hydrophobic, hydrogen bonding, pi stacking, salt bridge, and cation pi interactions, which can be seen in majority of drug receptor complex. So this graph is showing that there are around 65,000 interactions of hydrophobic type in the available structures in the protein data bank. Similarly, hydrogen bonding interactions can be found in around 30,000, and so on. So the hydrophobic interactions are observed when the two atoms interacting are approximately 4 angstroms apart. Hydrogen bond interactions can be observed when the two interacting atoms are approximately 3 angstroms apart. Now, these are approximate values. For example, hydrogen bonding interactions, they can range from 2.5 to 3.5 angstroms, but a typical average value would be 3 angstroms. Similarly, pi-pi stacking interactions, these interactions are observed in the two rings where the pi clouds are available, and those pi clouds can overlap over each other in order to exert a bonding effect. And then there are these salt bridges, which are typically long range but generally are having less than 4 angstroms length. So this is how the interactions can be perceived in molecular docking. So now let us come to the methodology of molecular docking. Let us just go through these two slides first, and then we'll see one by one what we can understand. So the first step in the molecular docking would be retrieval of protein structure from protein data bank, or if you have a homology model available for a protein, you can use that. Then we have to prepare our protein. Why so? Because majority of the experimental structures, they are deduced through the X-ray crystallographic techniques. Now the X-ray has a higher wavelength than the radius of hydrogen atom. So what happens in this scenario? The hydrogen atoms cannot be detected by the typical X-ray crystallographic method, and therefore the protein structures which are available to us in PDB, they are not having hydrogens. So we have to add hydrogens manually. Then we have to remove water molecules. These water molecules are again crystallographic water molecules which are necessary for the crystallization of protein. Now, these water molecules can interfere into the docking process, but some of the water molecules can be important if they are present in the active site and they are interacting or mediating the ligand and receptor interaction. Then, if there are some missing residues due to the resolution problems, we can add some missing residues so that the protein is prepared. Then the next step would be the preparation of ligands. So ligands have to be checked. Since they are the computational structures, we have to check that their bond length and bond angles are proper. So there are various structure minimization techniques which can be utilized, and checking and correcting partial charges. Then after these two steps, we have to create a grid. In a moment we'll see what a grid is, and then after the preparation of grid, we go to the typical docking process. So the first step is retrieval of protein structure from protein data bank. So this is a screenshot of protein data bank where you can search for a typical uh, receptor of your choice, and then after searching, for example, as I searched DPP4 and I have retrieved this structure called as 1x70. Now there may be multiple protein structures available for the same receptor. For example, if I talk about DPP4 enzyme, there are over 128 structures which are available in protein data bank. So it is also of pivotal importance how to find out a good structure for molecular docking. Remember, there's a phrase in computational drug design that is garbage in, garbage out. That is called as GIGO in abbreviation. So if you have a good protein structure, you will be able to have a good docking structure. If the protein structure is not good, then you can say that your docking would not be very much conducive. So how do you find out a good protein structure? So there are some typical uh, parameters. For example, the first is resolution. Resolution, it is the power of your crystallographic method to differentiate between two nearest particles. So for example, if you have a resolution of 2.1 angstroms, it means that your method will be able to differentiate, or it will be able to know that there are two different atoms present at 2.10 angstroms. Now suppose I increase the resolution and I say that there is a resolution of 3 angstroms. What will happen is that the method is able to differentiate two particles only if they are 3 angstroms apart. If they are near to one another, then it will consider that as one atom, and therefore the lower the resolution, the better the protein structure. So this is one parameter. The other parameter would be the organism from which you are deriving. So typically, if we are trying to develop a hit which is to be utilized for humans, you should go for the protein which is from the Homo sapiens origin. Mutations. In this case, you can see that there is one mutation, but typically, what you would like to do is you would like to go with some wild type proteins. Okay, so these are some of the parameters that should primarily be considered. The other parameters can be the R value and R value work, and a very good metric is given at the protein data bank, where you can see that all the, just a second. So this metric can give you a very good idea about the quality of a protein structure. So if majority of the parameters are on the blue side, you can consider that structure to be better than the structure which is having majority of parameters on the red side. Then we have protein preparation. So the first step would be adding hydrogens. Now adding hydrogens and removing waters you can use interchangeably. There's no issue. So as we have discussed that protein retrieved from protein data bank is not having hydrogen atoms in there. So we have to manually add these hydrogen atoms. So you can see the difference between these two structures. Here you can see that the hydrogens are represented as these white colored elements. Then removing water molecules is the next step. Here, these red dots you can see as the crystallographic water molecules. We can just remove these water molecules. Now, there are different programs we can utilize for removing these water molecules and adding up the hydrogens. You can use Pymol for visualization. You can use Autodoc tools. So these are some of the freely available softwares that you can utilize for such purposes. Then when you have done these two parts, then let us come to the next part, and that is the grid generation for the receptor. Now I have displayed this grid over this receptor, covering this whole receptor. Now grid is a place where the calculations for your docking are going to take place. So one can think that if I want to have a good docking, I can go for the whole receptor under the grid. But this is not a good technique. Why? Because it might be possible that you can find some interactions which are not there in the active site and they are off target. So what we have to do is we have to check the active site and we have to prepare a grid in such a manner that it is covering the active site only. So now as we can consider that this grid is built around the active site and it is ready for the calculations. Now wait for the types of calculations that we are going to see. Then we can go for the ligand preparation. So ligand preparation would involve checking the structure for bond length and bond angles. Suppose you are drawing some ligands in a 2D program or in a 3D visualization program, and then you want to check if this structure fulfills the basic criteria of bond lengths and bond angles and all. What you can do is you can just minimize this structure so that the optimum bond lengths and bond angles would be achieved to a certain extent. Now, how this is done, it is out of the scope of our talk. So we will just cease this discussion here. Then we can just check and correct if there are wrong partial charges assigned. Then we can check for total number of torsions. So torsions are something which are responsible for the movement of the molecules with respect to the bonds. We will see this in a moment. So what types of calculations will be done inside a grid? Remember, the grid is the space where you are going to dock your structure. So there are two things that take place in molecular docking. The first is conformational search for the ligand, and the second is scoring. Now let us try to understand in a nutshell what the conformational search is. So conformations are the various poses a molecule can take in three-dimensional space. So structural parameters of ligands such as torsional, that is dihedral, translational, and rotational degrees of freedom are incrementally modified. Now let us see what these torsional, translational, and rotational degrees of freedom can be. So if I just start this video, you can see that this molecule is rotating. So in a grid space, this molecule can take place any kind of rotation. It is not in our hands. Okay. So this is typical of a docking program. This is a translational movement. Okay. So the molecule is translating inside the grid box. And the third way would be the torsional degrees of freedom. Now, just wait to see how these torsional degrees of freedom can be considered. So if we have a torsion angle, and with respect to that torsion angle, we are able to rotate the bond, then we will be able to see some different conformations arising from this molecule, like the one that is coming. Okay. So if you see now, through this single bond, there is a rotation of this ring. So if you go by different frames, what you can see that in the first frame you have this ring here. Now the structure is same, but due to the rotation of this single bond, you can see that this ring is assuming different structures. And typically what is happening is the molecule is obtaining an entirely different structure from where we have started. So this is one thing that we have to have a very robust conformational search algorithm. So these algorithms can be of two types. One is systematic and the second is stochastic search. Then the second is scoring function. Now when we are playing this video, what will happen is that the molecule will rotate and it will find some interactions with the amino acid residues. The molecule will translate and it will find some interactions with amino acid residues. The molecule will have the torsion angles changed and it will have certain interactions with the amino acid residues. So each and every time you are getting a different kind of interaction with these amino acid residues. Which one is better? So the scoring functions play their role here. Okay, so there are four typical types of scoring functions: force field based, empirical based, and knowledge based. These three are typically called as the classical scoring functions. And the next is machine learning based, which are somewhat newer. So in systematic search, we can see that we are able to get a local minima very quickly. Now we will just very quickly skim through these slides so that we can understand the differences between these two. Okay, so in systematic search, you will change a conformation and then you will see what is the energy. What you want a conformation which is having an optimum amount of energy which can be obtained in a biological setting. So what we would want is always a lowest energy conformer. So in the systematic search, what you will do, you will change the conformation somewhat. You will check the score, you will change somewhat, you will check the score, you will change somewhat, you will check the score at every place. But there is a con in this systemic search, and that is it can converge into a local minimum rather than a global minimum. So what you are doing is you are taking only one structure, one conformation, and then you are trying to minimize it. So it might be possible that you can reach at a minimum energy conformation, but the actual minimum energy conformation is different. So the stochastic search comes to rescue here. What we are able to see is that in stochastic search, at the same time, there are multiple conformations generated and those conformations are minimized. Okay, so if the conformations are minimized, we want the minimum energy conformations. These minimized structures for the different conformations will be compared to each other, and the compound which is and the conformer which is having the lowest energy will be considered as the global minimum. So it might be possible that you can get a global minimum with stochastic search somewhat quickly. So there are some of the example programs which can use these stochastic search like Autodoc, Gold, ICM Doc, and the example programs for conformational search are Glide, Flex, Doc, Surflex Docs, Doc, etc. So this is about the stochastic. This is about the conformational search. Now let us come to the scoring functions. We will discuss the first three, that is classical scoring functions. The first is force field based. Now, as you can see in this equation, the force field based scoring functions will involve the interaction that are electrostatic or van der Waals in nature. So a compound will interact through electrostatic interaction or van der Waals interaction. Score will be calculated. The second conformation will go. The score will be calculated, and so on. The best score will be output. In the empirical scoring functions, what you will see is typically the energies are calculated and they are added in order to find the best score. Okay, so they can be van der Waals, the energy of van der Waals interaction, the energy of hydrogen bonds, and so on. The knowledge-based scoring functions are the scoring functions in which what you do is you have around 100, say for example, 100 protein ligand complex, and you get the interactions, the typical type of interactions in there, and then out of those 100 protein or 200 protein ligand complexes, you prepare an algorithm which is able to typically give the typical value of your score. And in this manner, you can utilize these three kinds of scoring functions. Since the time is less, we are not delving too deep into this, but I think this should be sufficient for beginners. So after the docking in the grid, what you will be able to see is the score. The score will come in the form of a docked pose and a docking score. Okay, so the docking score in this case, this is a typical autodock Vina docking score, which is displayed. Okay, so the lower this score, the better you can have the affinity of your drug to the receptor. So for example, minus 8.4 is the lower score. It is better than the minus 7.7 conformation. And a docked pose, remember we have discussed that a typical docking or a best docking would look like where the crystallographic ligand and the docked ligand will be overlapping each other completely. So in this docking program, I have used Autodock Vina. What you are able to see is that the blue is your crystallographic ligand and the green is your docked ligand. So what you are able to see is docked ligand is not completely overlapping on the crystallographic ligand, but it is very near to this. So in this case, what we will be able to see, we will be interested in if this pose is able to give us the same interactions those are given by the crystallographic ligand. And in this case, we are able to see that this ligand is able to give us the same interactions. So we can say that this docking is a good one, and the docked pose is fit very well into the active site cavity. And therefore we can say that the compound is docked very well. So this is about a very brief account of molecular docking. Now let us just in a moment see what is virtual screening. Virtual screening is nothing but the docking of multiple ligands or searching of multiple compounds over a pharmacophore. So since we are interested in structure based virtual screening, since molecular docking is the interaction of one ligand with a receptor at a time, in virtual screening, what you will be able to do is you will be able to dock more than one ligand, say hundreds of thousands of ligands in one go on the receptor, and among those you can get the best molecule. So I think this is it for this talk. So I thank you very much, Dr. Vivek Asati and ISF College of Pharmacy Moga for extending this opportunity to interact with you people. Thank you very much.
So thank you very much, Dr. Piyush ji, and I have listened your lecture, and I'm impressed you presented so many things related to talking, and I think our viewer get opportunity after viewing this lecture. So, sir, one of my question, only one question that,
Yes,
as you shown that there are different type of interaction that occur during the docking process,
yes.
and the main role is hydrogen bonding. So, why we just presented hydrogen bonding only, that not presented the hydrophobic and other interaction?
Uh, See, what happens, Dr. Asati, is that hydrogen bonding is considered as one of the typical, what we can say, non-covalent interactions, which are very easy to form and very easy to break. If they are there, it might be possible that your drug will be able to excrete from the receptor and in turn excrete from the body as well. If you talk about the hydrophobic interactions, what happens is that in the hydrophobic interactions, water molecules are also there. So these water molecules can interfere with the hydrophobic interactions to a large extent. We don't know in the actual setting of the biological environment what will be the consequence of this. But typical hydrogen bonding interactions can be very easily formed and easily broken. So I think, therefore, we are more interested in the hydrogen bonding or ionic interactions because they are very easy to form and break. So I think I'm able to give the answer to your question.
And presently, we listen a term covalent bonding. So,
Yes, yes.
what do you think that what is the role of covalent bonding in docking process?
So if we talk about covalent bonding, the major uh, drawback it was considered in the previous times was that if the covalent bonding is there, it might be possible that the drug receptor interaction will be perpetually present. But there are certain receptors where the covalent docking is giving very good results. And therefore, I think in today's scenario, covalent docking is as good as our normal docking that we generally consider as hydrogen bonding and hydrophobic or pi stacking interactions.
Okay, thank you, sir. Thank you. Very nice. Uh, okay, sir.
Thank you. Thank you.